Thanks very much, and thank you for the opportunity to come and speak. We've heard some fabulous presentations about what can be done with data science, with AI, with machine learning. But fundamentally, they need access to large, deep data. And that's really what UK Biobank was set up to do, to become a global resource for exactly that kind of application of novel methods to identify the causes and better ways to prevent and treat disease. In many ways, it's the kind of the Google Maps of health research, identifying the pathways from our lifestyle, our environment, our genes, to the development of disease, and finding better ways to travel through our lives. So as was mentioned, increasingly over the last decade, more and more researchers around the world have been using the UK Biobank data. We're always looking for ways in which to remove obstacles to access to these data, first in the UK, then the US, now in China, and many other countries around the world. But when I look at this graph, I look at the people who are not using the data. Those really smart scientists in less well-resourced settings. How do we make them have that opportunity to use these data? We've made it free for them to access it. We've made it free for them to compute on it. And we really want to encourage more and more great minds to use more and more expertise and techniques to find the ways to prevent and treat disease. But there are 4,000 projects going on. There are 20,000 researchers around the world using them. And the reason why I'm here is just to say, why aren't you using these data to help improve our lives? Every day, more than 10 peer-reviewed publications emerge based on the UK Biobank data. Every day. I mean, most scientists will publish, good scientists will publish 10 papers in a year. So the idea that we create resources and hold them to ourselves and say only we can use them is clearly dumb. The way to leverage the data and turn it into information that improves lives is by making it available. And again, we're seeing more and more publications, some 5,000 publications last year alone. But what exactly is UK Biobank? Well, it's the altruism of 500,000 middle-aged men and women from around the UK, selected from all social groups in all parts of the UK with different lifestyles, exposures, different genetics. And then we've asked them tons and tons of questions, made physical measurements of them, and most importantly, collected a whole range of biological samples that have been stored for the last 10 to 15 years, waiting for technology to catch up with epidemiology in order to be able to turn these samples in particular into data that can be used by scientists like you and others around the world. We have consent from all of these 500,000 participants to link to all of their medical and other health-related records, going back in time before they joined to understand about their health beforehand, but critically to be able to follow them, as Kate Bingham mentioned, is so readily done in the UK, by linking into their health records to find out what conditions they develop over time. So when I think about the dimensions of UK Biobank that make it so special, the obvious one is it's big. And it needs to be big because only a small proportion of a population of 500,000 will develop any particular disease. And so you need to have enough people at the beginning in order that enough of them will develop a particular disease that you have then the kind of reliable statistics to determine why it was that that person developed disease and another person didn't. But it's that combination of scale and depth that is particularly important. We need to know as much about the ways in which the participants have been living and continue to live, about the environments in which they live and the other exposures that impact on their risk of developing disease, and of course biological mechanisms like genetics, like the epigenetics that Steve Hawthorne talked about earlier, to try to understand the combination of these different risk factors for developing disease. And UK Biobank is not only very large, but it is uniquely deep. To the questions and the physical measurements that we recorded at baseline, we've been getting web questionnaires from participants to give us much more information about their diet, about their employment history. 100,000 of them have worn activity monitors for a week so that we can look at activity and sleep and determine the relevance of that to disease. 100,000 of them have now gone through a 5-hour imaging visit with magnetic resonance imaging of the brain, the heart, the body, the DEXA, so one can compare, as you saw earlier, how you can use DEXA and MR data. Cardiac monitors are going on them as well, so that we can look at cardiac rhythm, but also, again, at activity, at gait, and how that might relate to their brain images in terms of neurological conditions. And then we go back to those biological samples. About 20 years ago, the first human was sequenced. UK Biobank has sequenced all 500,000 of the UK Biobank participants. But it's not just about genetics, as Steve mentioned. We're also now measuring epigenetics, the changes in the genetics that are influenced by your environment. We've measured thousands of proteins using the new technologies that now exist to allow you to do that on tiny amounts of blood. 50,000 of those have been done, but now, during this year and next, we will complete thousands of proteins across the whole of the 500,000 cohort. One really interesting finding that came out very early, and the reason I particularly like this is this demonstrates in a number of ways the value of a large cohort, but also the value of making it available. This is the work initially of a PhD student in Cambridge, Massachusetts, that looked at genetic variants across the genome. You saw those Manhattan plots earlier in the day. And they demonstrated, contrary to everybody's prior hypothesis, that these genetic variants across the genome, most of which have very little effect on our lifestyle, our cholesterol, our disease rates, but you could combine certain of them and identify a few percent of the population that had risks of certain diseases equivalent to single gene disorders, but 10 times more commonly. So 3% of women who had a risk of breast cancer equivalent to the single gene disorders of BRCA1, BRCA2 that are only seen in 1/10 of 3%. Familial hypercholesterolemia, the most common genetic cause of disease. Again, 3% of the population at risk of a heart attack due to high cholesterol from these variants, equivalent to familial hypercholesterolemia, which is 1/10 as common. So one can see quite rapidly how this information could be used to make our screening strategies better. Rather than just screening for breast cancer at a particular age, we could be screening those few percent at a younger age. And of course, at the other extreme, the few percent that are very low risk might not be screened at all. In terms of giving cholesterol-lowering agents like statins, again, rather than determining that largely on age and gender, we could be using this genetic information to identify the few percent of people who would benefit from starting very early with a statin to prevent their arteries from furring up and avoiding them having premature heart attack. Most recently, using the protein data and applying AI and machine learning, it's been possible to allow the protein data to tell us what the disease the patients really have. So we're taking depression here, or researchers have taken depression here, and allowed the proteins which are part of the pathway from lifestyle and genetics to developing depression to actually demonstrate that depression is not a single condition. Again, a point that's been reiterated through the day, and can separate depression into different forms that are likely to require very different treatments. And this will help us to identify those treatments and ensure that we give the right treatment to the right patient with the appropriate condition. But when you take healthy individuals and you take a large number and you do lots of measurements in them, fortunately, you still have to wait. They remain healthy for a long time. And so these studies need to have very long follow-up in order for there to be enough people who have developed any particular condition. And that's where UK Biobank is at this point. We're seeing increasing numbers as the cohort ages. As our cohort ages, I am a participant in UK Biobank as well. And increasing numbers will develop various conditions, and that will allow scientists to identify why that is and hopefully find ways to prevent and to treat it better and to do so rapidly. So it is that combination of these 4 dimensions that makes UK Biobank unique. There are other large studies. There are other deep studies, but really none as large and as deep. And the problem with this is you can't go backwards in time. If you haven't done this 20 years ago, you have to wait 20 years. And so it was a prescience of the UK Medical Research Council government funding and the Wellcome Trust research charity funding, an idea to set this up at the beginning of the century that puts us in this position of being able to provide large, deep, and long data. But the most important aspect of UK Biobank, and the thing that the Wellcome Trust and the MRC required us to do, was to identify obstacles to making these data available, making them accessible. So now we have them sitting on a cloud-based platform with lots of tools to work on that, 30 petabytes of data, no way you could move it to 20,000 researchers around the world. So now the researchers can come to the platform and we can support them and, as I say, provide free compute to those researchers who need them, so that there are no real obstacles to using these data to make discoveries. And it is that combination of the 4 dimensions that is the key aspect. So it started with the Medical Research Council and Wellcome Trust. It's now supported by the major charities in cancer and heart disease and the NHS's research arm, the National Institute of Health Research. But it's also been really interesting in that we consider scientists wherever they are. This is not an academic exercise. Sorting out how to prevent and treat disease is not an academic exercise. It's an exercise that needs all the scientists wherever they are. And that any scientist in academia or industry can access these data. And what we found is that the companies are using the data and they're seeing the value of the data, and they are becoming funders of turning particularly the samples into more data to make it more valuable for all the researchers who need to use it. And indeed, increasingly, philanthropic funding to help ensure that the altruism of the 500,000 British men and women turns into improvements in all of our health. So we're dependent on your imagination, the imagination of everybody, everywhere, to make the most of this resource and find out how to treat all the conditions that will occur in these participants, in all of us, over the next 5, 10, 15, 20 years. Thank you very much.
Thank you, Rory. Great presentation. Obvious question, really, around the need to balance open scientific access to data with participant privacy and confidentiality. How do you balance those things?
I always think that there are two sins. The sin of omission and the sin of commission. And people always put so much emphasis on, if I did something, what would go wrong?
Yeah.
And I think we need to kind of rebalance and think, if I don't do something, what won't go right? And so I think it's really important that we have a balanced consideration of the opportunities to use these data to improve health. Yes, we have to make sure we protect the privacy of the participants, but we need, the safest thing to do is to allow no one to use the data. But it won't improve anyone's lives either.
Very similar, actually, answer from Reid Hoffman this morning regarding artificial intelligence. How do you think about ensuring that there's diversity representation within the biobank? How does that process work?
By setting them up in interestingly different places in the world.
Right.
What you want to do, I mean, the fact is none of us are normal, right? We have a lifestyle that is not normal. We have body mass index, which is not normal. So what you really want to do is be able to study the full range of lifestyles and exposures. You want populations where everybody is much slimmer, populations where people are much less slimmer, if that's a polite way of putting it. You want to be studying populations that have different rates of diseases. But you need to be able to follow people up. So you need to look to see where can you set up these resources. And so we've worked with the Chinese government and Chinese Academy of Medical Sciences. About the same time we set up UK Biobank, we set up the China Kadoorie Biobank of 500,000 people, all linked into their health insurance scheme. So I think just thinking about where are people interestingly different in terms of their lifestyle, their environment, their genetics, and their disease rates in order to increase the diversity that we're able to study and make those data accessible to the world's scientists.
And over the years, you've really kind of expanded into various areas. So there was the COVID and I think there was imaging as well, imaging studies. What directions are you planning in the future?
Well, in these kinds of studies, what you do is you tend to study the people in great depth, their lifestyle, their environment, their genetics, their proteins. But then you link into health record systems, which, as I think someone said earlier today, usually have a kind of, at best, 20th century diagnosis of disease. I think what we want to do now is take people who have developed particular conditions and really study that condition in depth. So people who are developing neurological diseases with funding from ASAP, the Parkinson's charity, we're going to invite them back and do imaging, biological samples, clinical assessment, so that we get a huge amount of depth on the disease to combine with a huge amount of depth on their exposures. But I also want to move away from disease to health, and the point that Steve Horwath is making about aging. So we'd like to bring everybody back so that we could study their health rather than just their disease. In order to understand what are the determinants if someone has a good trajectory in terms of health as compared with those who don't.
Rory, thank you so much for the presentation. Wonderful.
Thank you.